Peptides presented by MHC class II molecules are generated in acidified endocytic vesicles. Several classes of pathogen, including the protozoan parasite Leishmania and the mycobacteria that cause leprosy and tuberculosis, replicate inside intracellular vesicles and macrophages. Because they reside in membrane-bounded vesicles, the proteins of these pathogens are not accessible to proteasomes in the cytosol. Instead, after activation of the macrophage, proteins in vesicles are degraded by proteases within the vesicles into peptide fragments that bind to MHC class II molecules. In this way, they are delivered to the cell surface where they can be recognized by CD4 T cells. Extracellular pathogens and proteins that are internalized into endocytic vesicles are also processed by this pathway, and their peptides are presented to CD4 T cells. Most of what we know about protein processing in the endocytic pathway has come from experiments in which simple proteins are fed to macrophages and are taken up by endocytosis. In this way, the processing of added antigen can be quantified. Proteins that bind to surface immunoglobulin on B cells and are internalized by receptor-mediated endocytosis are processed by the same pathway. Proteins that enter cells through endocytosis become enclosed in endosomes, which become increasingly acidic as they progress into the interior of the cell, eventually fusing with lysosomes. The endosomes and lysosomes contain proteases, known as acid proteases, which are activated at low pH and eventually degrade the protein antigens contained in the vesicles. Larger particulate material internalized by phagocytosis or macropinocytosis can also be handled by this pathway of antigen processing. And that's peptides presented by MHC class II molecules are generated in acidified endocytic vesicles. Have a great day.